Subscribers, welcome back to AP Bio. Today we'll be working on 222.2, talking about the different types of speciation. So here we go. So yesterday we talked a little bit about how species can form, and now we're going to talk about the two different ways speciation can occur. One is called allopatric speciation and the other is sympatric speciation. Here's an image showing the two different types. So allopatric, if you think of like being patriotic, right? You're very drawn to your country. And if you remember like allosteric inhibitors, allo means different. So basically allopatric means a different country, speciation. This is when gene flow is interrupted due to a geographic isolation barrier. So basically genes can't move between a population because there's now a geographic barrier between them. And this happens when a population is geographically isolated from its parent population. And generally, this ends up leading to the different types of reproductive isolation we talked about in the last section. So allopatric speciation really depends on that geographic barrier, but what constitutes as a geographic barrier depends on whether the population can still stay together or disperse. So, for example, a canyon might be a barrier for small rodents, but birds that could fly over the canyon, coyotes that are able to make it across, or pollen that can float across and by the wind might not be affected by this geographic barrier. So what's a geographic barrier for one species may not be for others. Once the populations become separated, they can evolve independently through their own mutations, natural selection in that area on those mutations, and then, of course, genetic drift. And if the genes change too much, we may end up with a case of those reproductive isolation of any sort that we talked about in 22.1. So, for example, in the Bahamas, there's these mosquito fish that are found in different ponds, and they used to all be part of the same population, but within the different ponds, they evolved separately. So populations in ponds with high predation rates have a different body type than those in low predation ponds. The ones with lots of predators have a body shape that allows them to swim away really rapidly. But the ones that live without predators have a long, steady swimming body shape that allows them to kind of keep their body shape consistent. So those have evolved differently, even though they came from the same original population. They now have different body types due to the environment that they're in because they can't get from one pond to another. And so this individuals in each pond continue to interbreed with each other, and depending on which pond they're in, may be more or less fit. We also have been able to observe reproductive barriers that have been developed in a lab situation. So we can actually create experimental reproductive barriers to isolate populations and subject them to different conditions. So one lab experiment isolated two populations of fruit flies and gave them different food sources. And they eventually grew to prefer to mate with partners who were adapted to the same food source. So we had some flies that were raised with starch and some that were raised on maltose only to eat, and then we mated them. And we found that in the experimental group, those females that were raised on starch mated with more males that were raised on starch compared to those raised on maltose. And those females raised on maltose mated more with males raised on maltose. But in the control group, where they weren't separated, they were all fine to mate with whoever they wanted, like they originally could. We've also seen this in nature. So we observed some snapping shrimp that were sister species, but when they became isolated by the Isthmus of Panama, they were no longer able to interact with each other. So this tiny isthmus arose, which prevented them from crossing between these two, and they evolved to have different populations. Um, Therefore, we found that areas that have many geographic barriers, like lots of mountains or water barriers, things like that, have more species than regions with fewer barriers. And essentially, if you increase the geographic distance between species, that reproductive isolation increases. And just because they are physically separate doesn't mean that you can't necessarily reproduce. For example, we could reproduce with people living all over the world, but the reproductive isolation barriers can result from intrinsic changes to the genes, and if those populations are isolated and those genes have a higher fitness, those are going to survive and reproduce, and then eventually the genes may change so much that they can no longer reproduce. So, in contrast to allopatric, different country speciation, we also have sympatric, same country speciation. And this is when speciation takes place in a population that lives in the same area. So, there's no barrier. But for some reason, there's a reduced gene flow between the groups, even though they're still in contact with each other. So, there's a few different things that can cause this. One is polyploidy, one is habitat differentiation, and one is sexual selection. So this is basically where we get a new population from a new species and a new species, even though they live in the same environment as the parent population. So polyploidy is the first way this can happen. And this is when we get an extra set of chromosomes due to an accident during cell division. And you probably are thinking like, wait a second, how does that work? Well, this is very rare in animals because typically when we have errors in cell division, we can't produce a viable offspring. But we can see this a lot in plants. And basically, one species may have an error that results in them having more than two chromosome sets. So instead of diploid, they could be triploid or tetraploid and have multiple copies of chromosomes. And a lot of times, if you try to mate an autopolyploid with a diploid organism, like a regular organism, they might not be able to survive and reproduce very well. But sometimes, if you pair two polyploids together, they can survive and reproduce and produce more polyploids. So, for example, here we have a diploid cell which had 6 chromosomes, but now the tetraploid cell was created and we have 4N, a tetraploid cell. And when that cell goes through meiosis, we get 2 diploid cell gametes. But if those two can fertilize each other, we get this new tetraploid species. So there's lots of different plants that have been produced by polyploidy. Here's another one where you can see we've got our 2 species and we have the normal gametes, but this one and this one have they're 2 different species and they've come together. And this one has 3 gametes and this one has 2. And now we have this sterile hybrid because it has 5 chromosomes. But if we had another error where we doubled the chromosome number, now we have 10, and this species could be viable because the 10 can divide evenly. So this is what's called allopolyploidy. It's where you have polyploidy resulting from 2 different species coming together. So there's lots of crops that are polyploids, and we actually have plant geneticists that work together to produce new polyploid species in the lab, where they use chemicals to cause errors in cell division and create new polyploids. Sympatric speciation can also result from the appearance of new ecological niches in the habitat. So maybe introducing a new tree, and then all of a sudden these flies Some are going to be on the native hawthorn trees and some are going to be on the apple trees, and they may prefer to mate with those that have the same food source as them and eventually develop different traits. And then finally, sexual selection is a huge driver of sympatric speciation. So a lot of times certain organisms will only mate with those who have a specific color. And if they continue to mate that way, we may eventually develop a new species that is only wanting to mate with that color. So just a kind of a review, in allopatric speciation, it's a creation of a new species because there's actually a geographic barrier that prohibits genes from flowing between populations. And then that will ultimately potentially result in intrinsic barriers in the genes that don't allow them to reproduce with each other anymore. And eventually, even if they were to come back together, their genes would have evolved in two different directions that they could no longer reproduce. And in sympatric speciation, there's a reproductive barrier that isolates a population, even though they were never geographically separated. And the three most common causes of sympatric speciation are polyploidy, natural selection due to differentiated habitats, or sexual selection. So that's the end of this section. Hope you enjoyed, and don't forget to like,